just been ignoring. I'm just sitting there. I'm like, I know. I told her. I'm like, sitting there. I'm like. Yeah, I had a feeling. I was like, 500 bucks is fine. Take that or name. Went on with my evening like.
Are you all right?
Oh yeah, I'm just stretching my neck. I'm having like a really bad tension headache today, so I'm trying to get the neck stretches in. So well, we've got 9 people joined on. I'm gonna try to give people a little bit of a chance to join in and get their points for joining. I want to get started soon, so hopefully. Oh, it's just 4 of you guys here. Oh, right. We have some people online too. We have 11 people here.
From Norman when I left work.
It's gotta be more than that, right?
It's more than that, but it's not bad. Like, I'm not driving into traffic. And that's like I-35 and like 240 and come on.
Walk away. Norman, 16 miles.
I figured that right around it's true. Ooh. They go there on Saturday. Or a basketball game. Traveling. What side is Shawnee? My grandma works for Shawnee schools. So, okay, she works for Shawnee schools. That's about as far as I've always been. Like middle school to high school. She said she likes going to Shawnee High School. I mean, they got to stop that. Sexton, yeah, so he was my teacher when I was a freshman in high school. I think that's in high school. Coach Madonna. He's the one that left us and went on with it.
All right. So let's go ahead and get started with our Kahoot. Be sure that you have signed in if you haven't yet. So we've got 4 people here. We've got 13 people here, so that should be 17 people signed on Kahoot. So be sure you get signed in. We're going to get started now. Reviewing chapter 6. What is the term that refers to an organism's total sum of genetic material? All righty. Yep. Genome. This is just a matter of understanding the definition of that term. So moving on. Question 2. The physical expression of a genotype. Physical expression of a genotype. All right, so phenotype, one student in my other previous sections earlier this morning, she had said the way that she remembers that is that physical is pH, just like the pH that phenotype starts with. So thinking of it that way, if we have to, but again, this is just another being aware of that term. The genotype is literally like if you have genes in your gene that you have one from your mom, one from your dad, and they might be different from one another, but you're expressing one of those in your body physically. That physical expression of that and what we see in that person is the phenotype, or in any organism. The three parts to a DNA nucleotide. So this is specifically for DNA nucleotides. Alrighty, so we have deoxyribose sugar, phosphate, and a nitrogenous base. That's a DNA nucleotide. The green one down here, we have ribose, sulfate, and nitrogenous base. We don't have sulfate in there. For the red one, which is our next popular choice, ribose, phosphate, and nitrogenous. Remember, D in DNA stands for deoxyribose. So we have to have that deoxyribose in there. It can't just be the ribose. All right, so that's basically what that's going to be referring to. And that's why that one's the right answer. All right, DNA anti-parallel strands are two sides of our highway of our DNA. How are those strands of nucleotides linked together? All right, so most of us got this one, but this is going to be linking those things together. The A to the T and the G to the C are held together by attraction forces that involve a hydrogen. So this is hydrogen bonding. We mentioned that in chapter 2 back in the beginning of the semester. All right. True or false? DNA polymerase builds DNA in the 5' to 3' direction, adding on to the 3' end. Is that true or false? All right. So this one is true. I'm glad that we got that one. So it's always going to be in that 5' to 3' direction, adding on to that 3' tail every time with our nucleotides. So glad we were getting that one. Which enzyme connects the Okazaki fragments formed during DNA replication? Remember, in DNA replication, we have a leading strand and a lagging strand. That lagging strand is formed in fragments that we call Okazaki fragments. All righty, so we've got some disagreement here. So when we have DNA replication, that is literally the process of making copies of DNA molecules. We are not doing anything with RNA in DNA replication, all right? So we're never going to involve RNA anything in the DNA replication except for in one aspect of DNA replication. And it's going to come up in our discussion today in genetic engineering. And that is that our DNA polymerase can only add on to an existing strand. And so we use RNA primers to add our nucleotides onto. Those RNA primers are the only thing there that is going to consist of anything RNA, but they're going to get removed and deleted and replaced by DNA, because the whole process of DNA replication is literally just to make copies of the DNA molecules. That's it. So this is going to be DNA ligase. I don't know, I think I might have mentioned it in this class, but I don't remember, is that if you play a woodwind instrument, like a clarinet or a saxophone, the thing that holds your reed to your mouthpiece is called a ligature. So ligature brings those two things together and makes it into one. DNA ligase brings those two pieces of DNA together and makes it into one in those bonds. Those bonds are called phosphodiester bonds. Another L, yeah, another L in there. So during transcription, what happens? Alrighty, so during transcription, we're going from DNA to RNA. Transcription is a different process than replication. So the three processes that we learned about last time were replication, which is copying the DNA, transcription, which is going from DNA to the RNA working copy, and then translation, which is going from the RNA working copy and making it into protein. All right, what is a codon? A codon is 3 nucleotide bases of mRNA. All right, a codon is part of the mRNA. A codon codes for an amino acid, but a codon is not amino acids, right? So the codon is the 3 nucleotide sequence of the mRNA that the ribosome is going to help with reading and matching up with the anticodon on the tRNA that calls for one specific amino acid. All right, question 9. In what kind of operons do we have repressor proteins that are already bound to the operators unless we change the shape with the addition of another protein and remove it? All righty. So, and what kind of operons do we have repressor proteins that are already bound to the operators, that when something comes in, it causes them to unbind so we can actually get our product later? So what this is telling us is if the repressor is already bound, we are not making the product of that operator, right? So they're off. So in what kind of operons are we off that can then be removed that repressor and turn them on? That's inducible operons, all right? In repressible operons, they have a repressor. They both have repressors involved, but in repressible operons, they are on and the repressor will then bind and turn it off. So it's all about whether it is on or off when it starts, okay? So that's what that's about. So a lot of people getting that wrong. Just because it has a repressor in it doesn't make it repressible. Inducible operons also still operate on the idea of the repressor being there. It's just whether it was there originally or whether we're adding it later on to turn things off. All right, question 10, missense mutations. What are those? All right, so missense mutations. So we have 5 types of mutations really that we talked about. One of them was back mutations, which is just reverting back to the original. All right, so that was one of them. We had missense mutations where we have a nucleotide change that causes a different amino acid. That's a missense mutation. A nonsense mutation, we have a nucleoacid, one nucleotide base has been changed. One of those bases has been changed. That has led to us having now a stop codon. It does not code for any amino acid. And that is nonsense to us when we're making our protein product. So that nonsense mutations are stop codons that have now been introduced because of the mutation. So that's nonsense. So that's back, missense, nonsense. Silent mutations are if you have changed a nucleotide sequence, but you're still coding for the same amino acid because some codons still code for the same amino acid, right? So if that happens, that's a silent mutation. And then the other one that we talked about was the frame shift mutation, where you have addition or deletion of 1 or 2 nucleotides. That changes basically that whole reading frame of reading 3, 3, 3, 3. And if you remove 2, it doesn't skip the 2. It just starts again at the next one over. So it shifts everything over and then messes everything downstream up after that. So that would be the frame shift mutations. All righty. Podium. Good job, guys. Let's get out of that. And let's get into our lecture today, which is going to be over chapter 8. Some more DNA stuff here. Now we're going to be applying some of the stuff that we learned about from last time. All right, so real quick, I'm going to post. We might not get to, we're actually probably not going to get to questions yet again, another day where we're probably not going to get to questions, but the people that we have today for question 1, I have Carmina. For question 2, I have Jessica. You're still going to be responsible for your question if you're on Zoom, guys. Okay, for question, if we get to them, like I said, we probably won't. For question 5, we have Babylon. That's a 5 and not a percentage sign. I know you guys can't see what I'm typing, but I don't say it out loud. All right, and okay, so sorry, that was the question. Sure, we'll switch that. Who cares? I don't know how I messed up that order, but it's fine. Question 3 will be Tommy. And question 4 will be me. It's fine. Skipped around a little bit there, but you guys get the gist of it. All right, let's get started. So I put that in the chat if you guys are on that. All right, so recombinant DNA technology basically is utilizing DNA to either identify somebody, find underlying mutations so we can treat diseases potentially, or using technology like CRISPR or something like that to identify any sort of anomaly in the genetic sequence to then repair it. We can do that potentially in actual full living organisms that have already been born. So, all right, the whole concept of how this is going to be able to work depends on our understanding of DNA and how it operates, all right? So we have heating of DNA basically is going to release, whenever we had our double helix like that, where we had our GAT, so the Gattaca, we're gonna write that out, okay? So GCTAATGT, that these A's and T's are bound together by hydrogen bonds, as are our G's and C's. Okay, so those things are held together by hydrogen bonds. So just like we said on our question in the Kahoot, which most of us got that one, that our anti-parallel strands are basically held together by hydrogen bonds. And so we can heat those strands up and cause them to separate from one another completely. And that if we cool down that solution again, they will, because of chemistry, because A wants to bind with T and G wants to bind with C, that would be something that would allow those strands to anneal back with each other. Anneal means binding back together, okay? To stick back together because of our nucleotide sequence. So if you separate whole DNA strands with heat, whenever you cool them down, they'll bind back to each other where they're supposed to bind because chemistry favors that, okay? So this is a concept that's going to come back up later. So I know that we talked about there's only going to be a few instances of math in this class. And so we already introduced the fact that when we're looking at a microscope magnification, you're going to look at your objective lens times your ocular lens, and that will equal your total magnification. So I promised you that that should be easy math because that ocular lens is always 10. So all it is multiplying by 10. So we should be able to do that. But this is our next level of what I expect you guys to be able to do math-wise in this class, okay? So we have base pairs. All of our DNA is measured in base pairs. So if you have one strand of your DNA that said Gattaca, all right, just like the movie, if you don't know, then you should look into it because it talks a lot about the ethics of eugenics, which is altering people's DNA to favor certain traits, okay? So if we're talking about this being, again, 5 to 3 and 3 to 5, and this being your strand here, that if we were to write out one strand of it as being Gattaca, you know what the other strand says, right? and that this two pairs that we know that matches up with that G is the C, that this is one base pair. This is one base pair. This is one base pair. And so that this sequence that I've written here that spells out Gattaca is going to be 1, 2, 3, 4, 5, 6, 7 base pairs. That's the length of it. All right, so that's how that stuff is recognized. And we will basically look at different aspects of our DNA based on base pair size. And we'll compare things based on their base pair size. And this is not just for arbitrary reasons, but yeah. But whenever we start talking about like comparing, we look at CSI stuff, and I cut up your DNA with an enzyme that cuts at a certain place, and I cut up my DNA with an enzyme that cuts at certain places, our DNA will be different enough that we'll have different lengths of fragments that will result in that. And I can compare your fragments to my fragments in size without having to sequence the whole frickin' genome, looking just at those fragments and comparing them and their sizes to one another. Silence this real quick. All right, so that's the idea of why we're talking about this and why this is even relevant, okay? But, okay, so there's things that we need to know math-wise. One base pair refers to like that G that we know would be bound to a C on the other side, all right? If we talk about a kilo base pairs, or KB or KBP, either way, you might have that P and you might not. Kilo bases refers to 1,000 base pairs. And then a mega base pairs, so mega bases would be, you might have the P, 1 million. All right, so if I say that I have an organism that has, let's say, 4,000 base pairs in their gene, okay? And I have another gene that it has 7 kilobase pairs. Which one's bigger? That one, right? So it seems obvious at face value, but it's something that people get wrong all the time on the tests, okay? So just be aware that you know what base pair versus kilo versus mega means, so that if we have to ask a question on the test, which one is bigger, that you would be able to break it down and know the math behind that, okay? So that's another math thing that I expect you guys to be aware of. All right, getting into the theory behind some of the processes that are used in genetic engineering. CRISPR-Cas9. CRISPR and Cas9, these are essentially a set of enzymes and ability to recognize certain sequences that allow us to cut into DNA at certain sequences. So normally in bacteria, this is used to cut out foreign DNA. That's what it's for. All right, they have this as a natural system in their cells to be able to cut out DNA. And within this CRISPR-Cas9 system, it dictates that DNA sequence in how it reads it. So we can actually design CRISPR-Cas9 to cut at any sequence in DNA ever, and be able to cut into your DNA and cut out a bad sequence and potentially replace it with a good sequence in a living human being or any organism at all. This is obviously going to be much easier to do at a single cell level, where we can manipulate that cell and know all that organism has been taken care of, because that whole organism is just one cell. Whereas if you're talking about humans being millions of cells, how do you make sure that you made that edit in all of the cells? That's the biggest issue in genetic engineering when using CRISPR, getting it to all of the cells and actually having it affect all the cells. Right now, there have been no successful attempts at making edits in CRISPR in multicellular organisms that have stayed and been expressed the way that we expect them to so far. But you better believe there's lots of money going into this to try to fix any potential deadly or even harmful mutations that people might have. And instead of having to treat them repeatedly with a medication, actually just fix their genetic code for them. So that's something that is being used to determine that. So that's what CRISPR-Cas9 does. So we can potentially exploit the system to cut DNA anywhere and then potentially insert the proper DNA in its place. I'm not going to show the video today just to save time, but I recommend that the videos that we're not showing today, you guys take some time to go look at them to get some clarification on how these things work. It's something that is really a pretty hot topic in science right now. All right, the next thing is restriction endonucleases. These are kind of similar. So restriction endonucleases are enzymes that cut at certain sequences that are palindromic. We'll talk about what that means. To remove foreign DNA. That's what they're normally for in bacteria, normally. These guys are enzymes. We can't change the enzymes for these. The ones that exist are the ones that exist, and they cut at those sequences, period. So if we've been able to discover an enzyme that cuts at this sequence, that enzyme cuts at that sequence, period. So this is not as manipulatable. We can't change this one up to fit our needs like we could with CRISPR and Cas9. This guy already exists to cut only at that existing sequence. There's one enzyme that cuts at this sequence, one enzyme that cuts at a different sequence, and so on and so forth. So these sequences are always palindromic. And what does that mean? Just like race car reads the same forwards as it does backwards, okay? That's what that is, palindrome in the language of English, that these guys will do the same. So if we had GAA, TTC, and then the other strand would read GAA. Uh-oh, you starting to see how that works? TTC. So this reads GAA, TTC, and this reads GAA, TTC. Makes sense? So that's why these are palindromic, okay? Also, if you'll notice that if you were to fold these guys up down the middle, that the T would bind with the A's and the C's would bind with the G's. So they have an internal kind of palindromic aspect to them as well. So these guys are limited to having to match up like that, but this is how they recognize their sequences. So these are 4 to 10 base pairs that are going to be palindromic like this, and they always cut at a specific sequence. So one enzyme might cut at GAATTC, another one would cut at a different sequence, and so on and so forth. All right. So they recognize palindromes, which are identical when read from 5' to 3' on one strand, as they are 5' to 3' on the other strand. All right. So I'll draw that back up here so that we can see kind of what that would look like, just to remind everybody. So we'll do the GAA, TTC, CTT, A, E. These read the same thing, forward and backwards. This is 5. Okay. All right. So that's what that would look like on our actual strands. Okay. Okay. So what these guys do is they can, some of them cut just blunt ends like that. All right. That would create a cut with a blunt end that would just disrupt that genetic sequence, and it would no longer function as DNA that way. But another thing that it can do, most of these guys don't actually cut like that. Most of these guys, the way that they're going to cut is going to be like this. And so what that would result in is one side of that strand looking like C, and then G, A, T, T, and then the other side looking like T, T, A, G, C. So you get these sort of single strand overhangs. So this is what we call a sticky end. All right. So these guys want to bind up with the TTAA. They want to bind up with what they're complementary to. So when we cut them with these enzymes, they could rebind back with themselves. But now imagine that you have a sequence of, this is in a bacterial sequence that you've cut, and now you have a human sequence that you've cut at with the same enzyme, with the same overlapping sticky ends. You mix them together in excess, and now suddenly you can insert that human gene into that sticky end, and they'll want to stick in there. So that's how we're going to be using that. This picture of it to basically go along with my description. You cut out a bit of DNA here and opened it up with your sticky ends. Well, you can do that on 2 ends of a gene and stick it into a plasmid that you've also, remember, plasmids are circles of DNA that have extra genes for the bacteria. So we can manipulate those plasmids to insert human genes and then put the plasmid into bacteria and then force bacteria to express those human genes. Okay, we're getting how that will work. Okay, this comes back to the discussion that we had in the beginning about those restriction enzymes. Oh, sorry. About being able to cut people's sequences at certain places, and that if you cut mine up with the one enzyme that cuts at GAATTC, that you might have 4 fragments that result that are of this length, this length, this length, and this length. So they might separate out on a gel looking like something like this. This was Professor Yost, and this was William, and his might end up looking like something like this. He might have more. They might be of different sizes and that sort of stuff, just based because his genetic code is different than mine. He might have more GAATTC sequences than I would. Or maybe you guys have fewer. And so we can actually make these random cuts in your DNA and then be able to correlate that back. If you are an identical twin to somebody, you will have the same exact sequence as somebody else. Now, if we have a crime scene situation where we're trying to tell the difference between suspects at that crime scene, the odds of 2 people having the same little fragments being resulting from treatment with these enzymes is super low. I mean, we're talking like abysmally low, like it's extremely unlikely that that will ever happen if you are not identical twins. Even related people don't have the same fragments. So we can use the fact that we don't have to sequence your whole genome. We can just cut it up with an enzyme and look at our fragments that we get as a result of cutting it up. These differences in these little lengths and sizes of the fragments, the base pairs, this is called restriction fragment length polymorphisms, RFLPs, and you do need to know that acronym for the exam. So a restriction fragment length polymorphism, that term means that if you cut up someone's genetic sequence with a restriction endonuclease enzyme that cuts at whatever sequence it cuts at, in this case, let's say GAATTC, that the little fragments that you get, the lengths of them will differ from individual to individual, from organism to organism, okay? So that's restriction fragment length polymorphisms. Polymorphism just means that you get different results, all right? All right, so those enzymes cut at a specific recognition site, such as recognizing GAATTC or whatever it recognizes. And if a strand of DNA possesses that recognition site, it'll be cut into 2 pieces. So if my chromosome looks like this and it has a cut on that, then my chromosome suddenly looks like this. Then we'd have a fragment here and a fragment here or whatever, right? And when you compare the sizes of the chromosomes, now we've divided it up into parts, and they'll run out differently on a gel electrophoresis gel, which is something we're going to discuss here in a moment. So we can use basically what fragments you get from me cutting with this enzyme or that enzyme or that enzyme to determine a fingerprint of your DNA from that, and then compare it to somebody else's, right? So gel electrophoresis, that's what I had drawn for you in that picture of my sequences versus William's sequences, where if Professor Yost's looked like this, I don't know, whatever, and then Williams looked like this. But those are different from one another. What do those lines in that gel mean? This is stuff that we've seen on like CSI and things like that. A lot of us have seen what these gels look like, but many don't understand how it works. But guess what? In this class, you get to learn how it works, and that's part of the course, all right? So we get a readable pattern of DNA fragments. That's what the whole point is of gel electrophoresis. And what we do is we have a literal gel that has a similar consistency to the gel that we actually grow our organisms on. And it's called an agarose gel. Guess where it comes from? Agar. So it's a sugar from agar called agarose. And we use it to make a gel. And then what we'll do is at the very top, like let's imagine that where the Y and where the W is, that's where I'm going to put my DNA sample in. Then we're going to expose it to electricity. You guys remember how our backbone is repeating sugar phosphates, right? All the way down on both sides. And that we have the base there, that's the G or the C, and that those, or the A or the T, and those are bound together, right, with the hydrogen bonds. Well, because our backbones consist of a whole lot of phosphates, and phosphates we've already talked about have a negative charge, DNA has a negative charge. So opposites attract in chemistry. So when we run electricity through our agarose gel, there's a negative pole and there's a positive pole. Which pole do you think our DNA, which is negative, is going to be attracted to? The positive, right? So opposites are going to attract. So this will be attracted to that positive pole. So DNA is negative, so it runs toward positive. Electricity is being run through it, and we'll run to that. All right. All right. So the way that we get these different little sized bands, these bands to be different from one another, each of these is a different size. And they're not just going to be arbitrarily stacked in there and or by magic or because it's A's or T's or G's or C's. What these are basically separated out by is the literal number of base pairs in each of those fragments. Where at the top of our gel, top, we will have the larger ones. And at the bottom, we have a smaller. All right. And that's how they're going to separate out. This is because imagine that if I had some mesh that I had set out and I had different size of balls, the bigger balls are going to be less likely to pass through the mesh than the smaller balls, right? So the smaller things travel faster through the gel. They're not trapped as easily by the gel. So it's like walking through webbing or something. Or if you're at like a mosh pit, if you're at a mosh pit and you want to get to the front so you can see the band better, smaller people are going to be able to sneak through that crowd a lot more easily than maybe larger bodied people would be able to, right? Sneaking through and forcing your way through. The smaller guys have that advantage of being able to sneak through the gel faster as well. So the small guys travel faster, the large guys travel slower in the gel. And then that is what's going to dictate how those fragments move through the gel and create these images like we have seen before. So if we look at this image here, and we look at every single person's profile from all these different individuals, 1 through 8, take a look really quick at individual 3 and individual 4. They have almost the exact same profile compared to one another. They're probably, if they are in fact actual different individuals, identical twins. Okay, so this is where we can actually see if we have cut up someone's DNA into pieces and separated them out by size with electricity and using that gel to slow down the big pieces, and the small ones move faster, that we create these different bands that we can then visualize those different sizes and then compare each person's fingerprint. So here, when we look at this gel, which is going this way this time, right? It's on its side. But if we have evidence 1 and evidence 2, both of those evidences agree that there was an individual with this sort of profile that was at the crime scene. We found blood from these evidence things. This is the victim. So we know that this profile for this person is our suspect. Of the 2 people that we apprehended at the scene, who do you guys think committed the crime? Suspect 1 or suspect 2? 1, right. Because it matches our suspect's DNA here, right? And that's how we can look at that. So we didn't have to actually sequence that person's whole genome. We can just look at their DNA fingerprint. There's another problem. So there's that, and that's using to identify individuals. If we're trying to put our genes into other organisms and force those organisms to make copies of whatever the product is of our genes, bacteria cannot process our mRNA introns and exons. They don't have that machinery. They don't have that in their genes, so they don't have the machinery to process it. So if your genes look like this and have introns, which is the crap that we want to get rid of, so this is waste, we usually cut that crap out during processing, right? Via that process that we call splicing. So that's the term for that. We'll splice out introns and then join together exons. All right, and then when we join all those exons together, we have a fully processed, readable mRNA that is ready for our ribosome to translate it. So that's what happens in eukaryotic DNA only. But if you want a prokaryote, like a bacteria, to read that, you have to remove those for them. And so it's not easy to just remove those from somebody's DNA and make an mRNA. I mean, how do you just remove those from the DNA and join the DNA together? In the past, we used to be able to do that, and that's the only way that we had to deal with it. Now we don't have to do that because we discovered a virus called HIV that has an enzyme called reverse transcriptase that turns mRNA into DNA. And so that enzyme that we got from that virus lets us turn mRNA that I isolate from your cells that's already been processed and is already ready to go. It's got that poly A tail that our videos told us about, and we can isolate it based on having that poly A tail, and it's already been processed and ready to read by ribosomes, and just turn it into DNA. We call that DNA cDNA, which is complementary DNA. It's complementary to the processed mRNA. So we can turn that into a double-stranded cDNA and then put that into our bacteria. No introns included. But we can't just put your genes straight into the bacteria because of those introns, all right? So it's an important middle step that we need to remember has to happen if you want bacteria to make gene products from any eukaryote, all right? All right, if you're having to make all of these copies of this human gene, that you want all these copies of this human gene, we make insulin, for example. This is a gene for insulin. And what you want to do is ultimately, how do I click this? There we go, there we go, there we go. All right, put these genes into a big old plasmid that you're going to insert into bacteria, right? Well, you need a lot of copies of those genes and you need a lot of copies of the plasmid. But how do you get copies of those guys? This was where polymerase chain reaction comes in. We learn about PCR, which is what polymerase chain reaction is. PCR, yes, we're talking about the same PCR that we use to diagnose your COVID, all right? We learn about PCR as being a diagnostic tool. It is good for that too, but it actually has more application well beyond that. But all PCR is this polymerase chain reaction. The polymerase part of this is DNA polymerase, and it's a chain reaction because all we're doing is making copies of DNA. Does that sound familiar to anybody? Because this is a process that we learned about that happens naturally in the cell, right? And we call that DNA replication. So normally in our cells will copy DNA through DNA replication. This hijacks that same process to specifically make copies of certain parts of DNA. So we're not copying the whole genome with PCR, we're copying a specific sequence with PCR. So PCR targets a specific sequence that you already know of and makes copies of that sequence, all right? You can go from one DNA molecule to billions in just a few hours. You don't need billions to diagnose a disease. But if we're saying, I have little segments of DNA sequence that match up perfectly, we designed it perfectly just to align with recognizing if you have any DNA in your sample that lines up with the DNA for COVID, right, for the spike protein specifically. So we'll create primers that are usually like 16 to 30 base pairs that bind only to recognize certain genes. So what we're saying is that you've got a whole sequence of DNA in your nice little ladder, and this primer recognizes this portion here, right? So on the other side of the ladder, you would have another primer that recognizes this part so that you could make copies between this. This is what you'd actually be copying, is a sequence in between those two areas. Not the whole genome, just a sequence between your primers. And those primers are going to be designed to only bind to recognize sequence for COVID spike protein, flu, spike protein, flu. I'll talk about H1N1. The H or the N will have literal primers that will recognize that there is H there or that there is N there, for example. So these primers are designed to recognize whether or not you have that gene there. So you have to have the primers that are designed for that gene, and you have to know what the sequence is in order to make those primers. So if I swab your nose and you're sick with some sort of respiratory disease and I swab your nose, I don't just put it into a little tube and say, hi, PCR machine, tell me what disease this is. It doesn't work that way. You say, hi, PCR machine, I'm giving you some primers for the COVID genes. Tell me if there's COVID genes in my sample. That's what you're asking the PCR machine to do. All right. So PCR doesn't diagnose for you. The PCR machine just tells you if you have the gene that the primers match with. That's it. So if you don't have primers to the disease that you're looking for in there, you will not know if that is there. Also, you can't just mix it up in one tube. Because if I'm running one PCR cycle that will be able to have primers for COVID, that's only going to tell you if you have COVID. It ain't going to tell you if you have tuberculosis, if you have flu, if you have pertussis or diphtheria or RSV or common cold or whatever. It's only going to tell you if you're positive or negative for having DNA that represents COVID, period. Okay, so that's what PCR is going to do. It literally only makes copies of DNA. And so if you do not have COVID and I give you the patient sample, the primers for COVID, and we're saying, hey, make copies of COVID DNA that's in the sample. If you don't have it, you don't make product, you're negative for COVID. If you have it, you make the product, you're positive for COVID. That's literally, it is way more basic than what you guys probably thought it was. It's hella basic. It's literally just asking, if I give you starters for COVID genes, does this sample have COVID in it? So now we're starting to see if somebody shows up to your clinic that has a less common disease and you have a PCR machine and you don't have primers for that gene sequence for those diseases, you can't use PCR to diagnose that. You can use other PCR maybe to rule out those other things, but how do you actually diagnose those other diseases? And that's where those tests that we are learning about in the lab come into play, because not everything can be tested with PCR because you don't have the primers for everything. God help you if somebody shows up to your clinic with the plague, right? You're not going to have PCR primers for the plague. Even freaking Baptist doesn't have that. People just don't come in with the plague very often. You might be like, yeah, because the plague doesn't exist. Congratulations, you're wrong about that. We have that here in the United States. Prairie dogs carry the plague. So you get a tick bite from a tick or a flea bite from a flea, that bit of prairie dog that carries the plague, and you are a hiker and you go hiking in areas where there's prairie dogs that carry the plague, which is very common in the Four Corners area of the United States, that you might end up with the plague as a result. And if you go to a hospital, they aren't going to test you at that local hospital. They're going to have to send your sample off to the CDC or to Los Alamos, where they will do the PCR reaction. So y'all going to have to wait for the shipping and everything like that. Meanwhile, you're sitting there with bubonic plague, hoping that you don't die, right? So we can't only rely on PCR to diagnose everything. It doesn't work that way. You have to know your primers. The primers are specific for that gene. All right, so we use different primers for recognizing COVID versus primers that recognize flu versus whatever. Okay. All right, so how does PCR literally work? Well, it actually works very, very similar to DNA replication. All right, so what we have to go for first is taking your ladder and then separating the strands. We get one strand separated out and one strand separated out. And then just like what we had with our DNA replication, we'll make copies of each strand, old and new, half of it, right? But first we have to have our primers bind. So that might be where the primers bind. Come on, guy. And then we'll build off of those primers, because just like with DNA replication, we have to build off of something. Can't build off of nothing. And that's what the primers are here for also. So our primers in this case for PCR are there to help recognize the specific gene if it's there or not, but they're also there to help our DNA polymerase build off of. So they're providing that framework for it to add on nucleotides for, okay? So literally we're just going to separate the strands, attach our primers, and then build off of the primers to create that complementary sequence in nucleotides. And then after we get that built, what are we going to do? Repeat and repeat and repeat. So the things that are involved, we have those primers. Again, they're going to be 15 to 30 base pairs. That's going to recognize a specific gene. You have DNA polymerase, same old guy, adds complementary nucleotides to the end of a primer. To the 3' end of the extending strand, right? Starting with the primer. All right, so those are the same enzymes that we use for replicating our DNA. The problem is that we're going to be heating our strands up over and over and heating and then cooling and then heating and then cooling and then heating and then cooling over and over and over again. And the heating that we need in order to actually get proper separation of our strands for this to work is very high heat. We're talking around 92 degrees Celsius. This is very close to boiling temperatures. The reason that boiling kills things is because it denatures proteins. So the proteins, when we get that nice happy little bow that gets tied because of all of those hydrophobic or hydrophilic reactions that are going inside and outside of our nice bow that we tied with our protein, the reason we have that is because of our hydrogen bonds. And so those hydrogen bonds are going to get disrupted from the heat, and it's going to denature our protein along with our DNA. So what we have to do is use high temperature things that are basically resistant to high temperature for this. And so we have isolated DNA polymerases from bacteria that live in the we call them thermophilic bacteria. They like the high temperatures. These are guys that live in like the deep sea vents, like where it's hotter than boiling. If their DNA polymerase can survive that, then they can survive PCR easy. So we use their DNA polymerase to drive this reaction, which is why PCR isn't cheap. Okay. Here's how this works in our nice little happy little image here. Let's see if I can zoom in on it. Maybe. Here we go. Okay. So on here, we will have our double strand. We will first step, denature. So here we're talking about 94 degrees Celsius, very close to boiling. Heat separates the strands. That's denaturation. Second step, priming. We'll cool it a little bit so that your primers, which will be in excess, very high excess. So when we cool it down, these primers will bind on. That's our second step. This priming step is also known as annealing. And then our third step is called extension. Extension is just adding on complementary nucleotides from the end of those primers, all right? And then after that, we're just going to repeat that over and over and over again. Literally the same 3 steps. The product that you made in step 3 will separate that, and then add primers again and build off of it again, and continue that over and over and over again. All right, so basically what we have there is that in your first step of PCR, in your first cycle, cycle 1, why is that doing that? You will basically go from 1 DNA strand and you'll make 2. In cycle 2, you'll go from, I don't know why it's doing it that way, but whatever, to that. So in cycle 1, you've gone from 1 to 2. In cycle 2, you've gone from 2 to 4. How many would we have in cycle 3? 8. And how many in cycle 4? 16. Yeah, it really is that easy. So you're just going to double every single time. All right. There will be a question on the lab exam that will ask you that. I'll say if you start with 1 piece of DNA, how many would you have after this many cycles? I'm not going to ask you 452 cycles. I'm going to ask you like 5 cycles. Okay. So being able to know, to write it out, hey, if I went through 1 cycle, I will then have 2. So the second cycle, if you have to write it out on your paper, scratch paper, then do it that way. Okay. Some people literally draw out the picture too, and that's fine also. All right, so those are our 3 basic steps of PCR: denaturation, priming, or the other word for that was annealing, A-N-N-E-A-L. I don't know why it's doing that, but that's the same thing. All right, so we're gonna move on. There's a video for this one that I encourage you guys to watch. Okay, so we're gonna use all of that crap that we just talked about in order to basically create organisms to express the genes that we want them to express. These guys are going to be called genetic clones. So if I get a bacterial cell that I have a modified plasmid that I've inserted a human gene into, and then I put that plasmid, take this, put our human gene in there, put this into the cell, and now that cell is going to multiply out to like millions and millions of cells like overnight. We already know those suckers really just like copy themselves. And so now those are our genetic clones. Okay. Cloning is the process of getting this done. So you're selecting a gene from an animal or a plant or something else. That is the genetic donor. So we have a gene from a genetic donor that we'll insert into a vector. This is typically going to be a plasmid or a virus. We can also use artificial chromosomes. This is literally chromosomes that we designed in a lab. So you can use any of these three things. And then you'll insert the plasmid that you designed, your virus with genes that you put into the virus. Instead of it having viral genes, now it has whatever gene you're interested in, or your artificial chromosome. And we're going to put that into our cloning host. This is usually a bacterial or a yeast cell because these guys grow so fast. And they're going to crank out the products. But it could be any kind of organism, all right? So strategies that are going to be involved in this: removing DNA from the host cells, synthesizing mRNA transcripts that are already processed to generate complementary DNA using reverse transcriptase, or using PCR to amplify genes or DNA that we've gotten. Or if you take mRNA out of a cell, turn it into complementary DNA, you might have only got a little bit from that process. We can put it into a PCR machine to amplify it. You'll make a ton of copies from it. The PCR machine will just make copies. That's all PCR does, right? So we can do all of these things and use all the technology that we just talked about to build that stuff. Remember that a plasmid is a circular bit of DNA that has extra genes on it. This might be a gene here, and this might be a gene here. And that these extra genes, usually in nature, will help with survival of the bacteria somehow. Might impart some sort of antibiotic resistance or toxin production or something like that. So little small circular bits of DNA. We will utilize ones that we understand well and that we have engineered the genes on in order to insert our copies. I'm going to show you an example in a moment. Another thing you can do is use bacteriophages. You guys remember when we talked about lysogenic conversion? Getting new gene traits from having been infected by a phage that inserts its prophage DNA into the bacterial genome, right? So that is lysogenic conversion, but we can hijack that system to have the phage introduce our gene of choice. And that process is called transduction, which we learned about in the previous chapter, right? Using the bacteriophage. So this is an image of a plasmid. And that's why it's called P. This means plasmid. So this is one that we call PUC19 that is actually very, very well characterized, very well controlled. It has the origin of replication. This is just the site where we're going to start with our DNA replication when we replicate the cell, where we start our replication forks. That's all that is, okay? Then we have two genes. We have this gene, which is AMP-R, which is going to be ampicillin resistance gene. In case it isn't obvious to you guys, ampicillin is an antibiotic. So this is a gene that we have in this plasmid that's already built in with resistance to ampicillin. So now, if I'm trying to tell you that we would have your bacterial cell that looks like this, you have your tiny little plasmid, and you want to know that for sure you've got your plasmid inside of your cell, you're going to grow your cells on a media that has ampicillin in it. Because if your cells, which were also very well characterized, they're not just wild in cells off of the street. These are cells that you're buying from companies that are well characterized. And you know that the cells that you bought from Fisher or Thermo or wherever you bought them from, that those cells are going to be susceptible to ampicillin. The only way that they're going to be able to grow on media that you added ampicillin to is if they have an ampicillin resistance gene. And the only way they could have gotten that is because they took up your plasmid. So now we can know for sure that the bacteria that are growing on your plate with ampicillin in it have to have that plasmid in it. So you can already control for that without having to sequence it. Okay? The fact that it grew means that it has the genes. All right, the other thing that we can do is insert, this is another gene that does something like breaking down lactose or something. And that we can interrupt that gene by inserting your gene of choice by using a restriction enzyme. So this particular site is called a multiple cloning site that can be cut with any of these restriction enzymes. So this could be GAATTC, which we talked about earlier, which is cut by an enzyme called EcoR1, or any of these other things. It has all these programmed into it. If you insert a human gene or any other kind of gene into that LAC-ZA gene, it's going to disrupt that LAC-ZA gene's ability to function. And now it can't do what it was supposed to do. And that's another way you can know that your gene got inserted into that, because that original gene got disrupted, number one. Number two, the fact that it grows on ampicillin media means that you have the plasmid in there. So you have controls of knowing that your plasmid got into the cell and that your plasmid contains the gene that you're trying to insert into it. Okay? All right. When you are taking a plasmid and sticking a gene into it, whether you have sticky ends or not, this stuff doesn't automatically seal itself up naturally. Just like our Okazaki fragments didn't seal themselves up together, you need DNA ligase in order to do that. So DNA ligase seals our sticky ends. Those bonds, or those phosphate sugar bonds, are called phosphodiester bonds. So ligase repairs those. The other thing that we might need to use is reverse transcriptase, and that's taking that mRNA that has been processed and turning it back into the DNA. DNA is what you have to insert into the plasmid. Plasmid is DNA. So you can't put RNA in there. You got to turn it into DNA. You need it to be processed. No introns. So we have to use that processed mRNA, turn it into DNA, and then seal that in. So there's a lot of steps involved, but it's bringing all that stuff together, okay? So here's the steps of how that would all work. You get your isolated gene from your genetic donor. So that could be a healthy insulin gene. Healthy insulin gene from somebody that makes insulin properly, and you're going to take your plasmid. Now we're going to cut both of those, the donor gene and the plasmid. We're going to cut them with the same enzyme. This one happens to be EcoR1. You don't need to memorize those, but that's the GAATTC guy. And so we're cutting those. We're creating our sticky ends for the plasmid as well as this. And if we have enough excess of your donor DNA, then most of your products will have plasmid stuck with the donor DNA. Then we can force the bacterial cell to take up that stuff. Okay. Then we'll grow up that cell. And while those cells are growing, they will go from the typical process that we learned about in the last chapter, make your mRNA transcript from your DNA, and then process it into protein through translation. All right, remember that. And then the protein will get exuded into the media, and then we can filter that protein out of the media and then put it into syringes that you can now shoot yourself in the belly or whatever it is where you shoot your insulin into your body so you can give yourself insulin whenever you can't make it yourself because you have a genetic defect for it. Okay. I bring up insulin a lot because it was one of the very, very first ones that was ever done this way. All right, so nowadays, if you get insulin, it's made because of this, okay? All right, so based on how we're making our cuts in our genome and those little bands that we get in our gels, we can make maps of your DNA fingerprints. We can also use the entire sequence of your genome to determine essentially what your genome looks like versus another person's genome. And now we can learn more about genes and what your genes are actually coding for and what a normal gene looks like versus a non-normal gene, what a mutation would look like, and predict the effects of those things. So this is basically genome analysis. We can also create nucleic acid hybridization probes. And so basically what this means is that if your sequence reads GAATTCGCC something, whatever, and that this would be normal. And you have a patient with breast cancer, and that maybe theirs looks like GTTTTCGAA. So you've got some problems here. You can make a probe that looks like the complement to that, which would be CAAGCTT. If this matches the sequence of the mutation, it'll stick onto it. And that this is called nucleic acid hybridization. Nucleic acid hybridization. What I'm saying is that I can get samples from a breast biopsy, get the DNA out of it, and just mix it with a fluorescently labeled probe. And if this DNA sequence sticks to your patient's DNA, that means your patient has the mutation. I don't have to sequence their whole genome. I can literally just see if my sequence sticks into their cells. And that is nucleic acid hybridization, all right? So that thing here that we have, right, that we just made here that is labeled with a fluorescent probe. We know what fluorescent means. It just means it glows a color, right? So if we have that fluorescent probe there, that allows us to visualize it without having to sequence it. So this is a gene probe. That would be an example of a gene probe. It's a short stretch of DNA of a known sequence that we can use to probe your cells to see if you also have the matching sequence, the complementary sequence. We can use enzymes or fluorescent things attached to them to see if you have it or not. We can use that to diagnose like cancers or mutations, as well as whether or not you have genes from tuberculosis in your sample, right? And this is just an image that is depicting essentially how that would work from step to step. I'm not going to go over it, but it does the same process of what we're talking about, using the probe, taking cell sample and seeing if that probe gene sequence matches the gene sequence, and you get binding that way and the fluorescence will stick. If you're doing this in somebody's like breast tissue biopsy, right? So I get an actual biopsy from their cells. I match my probe that has mutation in it. If it binds to that person's cells and makes them fluoresce, this is fluorescent in situ hybridization, or FISH. So I don't have to kill the person, and I don't even really have to kill their cells. I can literally just take that sample of their biopsy and see if my probes bind to that actual sample and look at it in a microscope. If there's glowing, my sample bound and you have a mutation. We can even take whole entire trays where all that these trays have is a mini probe. Here, a different mini probe here, a different mini probe here, and each of these little probes have a different sequence for a different kind of cancer, or a different kind of bacteria, or a different kind of mutation for some other thing, okay? So each of these little probes will be laid out on like 364 different probes, or you might have duplicates, of course, just to verify your results. And each of those probes, I will now take the patient's DNA that I've extracted from their cells. I take your biopsy from your breast tissue. I break it open. I take your DNA and I mix it onto this tray. And then we have lasers that will tell me if that patient's DNA bound to that spot or not. If your patient's DNA bound to that spot, you have the mutation indicative of that spot. So that might be a certain kind of breast cancer or a certain kind of colon cancer or something like that. Okay. So now applying the fact that we have known mutations that exist for certain kinds of cancer that we can test for if we know what they are that way. So this is DNA microarray. All right, we already talked about DNA profiling using the fingerprints, creating the different lengths of DNA using cutting. All right, and then we also mentioned briefly basically making sequence maps. So mapping out the whole genome and what genes you have in which order and what a normal gene would look like versus different variations. For example, eye color. Brown, green, blue, gray, and all the variations of those things. You have a whole lot of different versions of a gene that we don't consider them to be one is normal and these are all abnormal, but they're all versions. And so knowing the different versions of those genes versus potential mutations of other genes and things like that creates sequence maps of organisms that we can then predict what those things would look like if we had anomalies. And so we can annotate the entire genome by understanding what your genes are supposed to code for and what the code is supposed to say. We have also learned that if you have a genome where your codon says AAA and you're supposed to be making phenylalanine, and that gets changed into, I don't know all of them, so please give me a break here, but if we're making this up, right? So it gets changed, that A gets changed into a U, and now you have a codon that codes for lysine instead. That's a different amino acid, and this can drastically change how your protein folds. Instead of folding like that, now it looks like something like this. That protein's not going to function like it normally would because we have a different type of amino acid there, potentially, right? So it's how we get different changes from genes to genes, but it's also how you can get mutations that can cause problems with health as well. So having a single nucleotide change just from AAA to AAU can cause a drastic change in the protein and therefore the function of that protein. That could be potentially deadly, or it could even cause issues with how that protein just functions in general. This is called a single nucleotide polymorphism. So that single nucleotide can change and then cause this effect. So for example, in thrombophilia, I have the genes up here. Normal DNA sequence would have a GCT here. Instead, they have changed one nucleotide to GTT. Instead of having arginine, now we have glutamine. Glutamine causes a different folding in the protein that now causes excessive clotting. And now you are at an extremely high risk for stroke in your life. So you have to be on medications to control your clotting if you want to survive. So this is one nucleotide change in that person's whole genome that their life now is in detriment because of that one nucleotide change. So this is an example of a single nucleotide polymorphism that leads to a detrimental effect in multicellular organisms like humans. We already talked about microarrays, so I'm not going to touch on that again. We can use recombinant organisms to make products like medications. We've already talked about making insulin, for example, which is one of the first ones that we have. Human growth hormone is manufactured in a similar way. And we can even take corn and put genes that are, take normal corn, put a gene from a bacteria that makes that corn now resistant to being eaten by insects. So now we don't have to treat our corn with pesticides that could be carcinogenic to people. And now the corn just naturally doesn't get eaten up by insects as much. And so that process, these are just some examples of some other things that we make with recombinant technology, just FYI. That's what all these slides are. That process would generate a genetically modified organism. That corn would then carry genes from a microbe that help it be resistant to being eaten up by insects. That's a GMO. A GMO, or a transgenic organism, they're the same thing. GMOs are transgenic organisms that all they have is that we have put in another organism's DNA into that other organism. Now, if you eat corn that has genes from bacteria in it, that would make it a GMO. And almost all of the corn that you eat in your whole life is that, by the way. Almost everything that you eat here in America is probably GMO to some extent. It's hard to find things that aren't anymore, right? Either we have genetically engineered them through this process, or somebody has done gene mixing through the traditional way of just crossbreeding things. Okay, so most of the things you're going to eat are GMO. And a lot of people, when they're learning in this class, they're like, GMOs are bad. And they have it in their mind from these conspiracy theorists that have spread to our minds, thinking that if you eat a corn that has bacteria genes, those bacteria genes can then jump into your genome and cause you to have problems in your genome. And now you have weird foreign genes that are false, absolutely actually ridiculous if you know anything about DNA. Guys, if you eat just normal corn and you eat up that normal corn, your body is going to digest it down, break up the DNA, and use its DNA parts to make new DNA for you. You break it down, right? If you ate just the bacteria, your body's going to break it down, break down the DNA, and use the parts of the DNA to build new parts for your cells. It's not incorporating that foreign DNA just because you've mixed the corn and the bacteria DNA together. That is absolutely ridiculous. So if you know anything about DNA and how DNA works, and that we're using single nucleotides to build our own DNA, your body's going to break it down regardless of if you mix two organisms' genes together. 0% chance of that happening. I want to say that. It's absurd to think that it would. And I'm sorry if you like believed that and now you're telling me I'm absurd. Yeah, I am telling you that you're absurd for believing that, because that's not how life or science works. You're eating other organisms all the time. Whether they're GMO or not, your body's going to break down their DNA into its tiny little components. There's no risk of that gene jumping into your genes. But is there a problem with GMOs? Absolutely there is. Why? Because corn can transmit its genes to other plants. It has the ability to have its genes be able to jump to other plants. Plants don't work the same way as us. They can share their DNA with other plants. So through the soil or through other insects or through other means, they can get now this anti-insect bacteria gene transmitted to wild plants. And suddenly now you've disrupted an entire ecosystem that those insects depended on those plants for life. And now they can't grow because we have anti-insect genes introduced into the wild because you introduced them into crops. That's where the problem comes from, because plants can switch their genes up like that. Humans cannot do that. All right. So there's 0% risk of you eating a GMO product or putting a GMO product on your body, of you getting the genes incorporated into your genome, 0% risk of that. But there is a risk of the plants potentially transmitting those genes to other plants, or bacteria transmitting those genes to other bacteria. There is risk of that. Okay. That's the problem with GMOs. There is a problem with GMOs. It's just probably not what social media would have you think. Okay. We'll put it that way. All righty. So this will lead to development of things like super weeds or resistance to things and whatever. And we are almost done, I promise. All right. Makes sense now? We good on that? So now we don't have to use pesticides as much, and that's great. And it's great for the farmers too. But it's just a matter of the risk that we might be putting on the rest of the environment. And so how do you weigh those things and what's good or what's not? All right. So now we're moving on to gene therapy, which is our last topic. Repairing a faulty gene in an organism that's already been genetically determined. So when your mama and your daddy's genes came together to make you as an organism, now your genes are set for life. Except now I'm telling you they're not. They don't have to be. I can literally introduce new sequences into your cells to repair anything that's wrong with your genes. This isn't just used willy-nilly though. We only have a very, very few diseases that we can treat with gene therapy in a multicellular organism that's already established. So essentially how this is going to be working is take a normal gene from a healthy person that's already making the thing and clone it. Now we're going to insert it into a virus. What kind of viruses do we like to use for this sorts of thing? Well, I'm glad you asked. HIV. We've already told you that HIV inserts its genes into your genes. So we're going to hijack that. We're going to carve out all the HIV part of it and make HIV insert this gene into your cells instead. Suddenly now we've got a blessing in disguise of HIV that's not only given us reverse transcriptase to make cloning easier, but it's also going to insert proper working genes into your cells for us. We can hijack that and force it to insert good genes in. So we take HIV, we plug in, take out the bad genes, plug in our good gene. Take out the virus stuff that makes it virus and plug in our good gene. And now instead of inserting HIV genes into your cells, it's inserting this healthy working gene into your cells. We'll take that patient's bone marrow, mix it with the virus and infect their bone marrow with our engineered virus, and then infuse that bone marrow back into them, and it'll get established as part of their normal body again, and then copy those cells. And those are stem cells. So other blood cells after that, all the progenitors from that will be carrying those genes. We have successfully used this to treat spinal muscular atrophy, inherited vision loss, and acute lymphoblastic anemia. Okay. This is going to treat the cells that are in your body that are not your gene, like your gametes. So this is your non-sex cells only. It doesn't treat your sex cells. Your sex cells are kind of housed away. They do their own thing in a different organ in your body. They're not going to be affected by this. So this is not heritable. The hope is that then we could then apply this same theory in a germline way. And a germline is heritable, but we're going to be changing the egg, the sperm, or the early embryo. So if you're doing something like IVF, we can sequence your IVF embryo's genome, see if you have any mutations in there, and repair them before they turn into a whole ass baby. And then that baby now has that repaired gene in there. Now, we can see that that would be very beneficial if we're looking at potentially deadly genes. If you are having an issue in family genetics where your babies are consistently having some sort of genetic defect that is leading to loss of the baby, or potentially that baby growing up to having a deadly disease that would be harmful to it or just something that's detrimental to them, that we can catch that early on in that IVF process. But it also opens the window for designer babies. Choosing that your baby has blue eyes instead of brown, choosing that your baby has blonde hair instead of black, choosing. And now suddenly the people that have the money can design their own babies, and those babies will be perfectly designed to succeed in society, whereas people that don't have the money will have babies that are just naturally born, and they will not be as well equipped to succeed in society. So there's obviously ethical concerns in that. That whole process is called eugenics. If you don't know what eugenics is, look into Nazis, because they tried to do this. They were trying to basically breed in desirable traits of what they thought were Aryan traits and eliminate traits that they thought were Semitic. So you can see that there's a problem with favoring certain traits over others based on just favoritism, based on racism, or based on societal just having more money than somebody else. So that's where that movie, if you haven't seen it, Gattaca comes in. It touches on a lot of that stuff. We have a guy that was naturally born versus his brother who was engineered, and his brother is trying to track down this naturally born guy that's trying to fit into this astronaut program. And it turns out it's his brother the whole time. And it's Ethan Hawke and Jude Law and Uma Thurman. If you haven't seen it, it's a great movie, and it touches really well on this stuff. It's from like the 90s or something, so it's a little dated, but the ideology behind it still works. All right, so our questions, just to review this stuff. What type of sequence do restriction endonucleases cut at? This is palindromes. What does PCR do? Just make copies of DNA. That's it. Gel electrophoresis. Which pole do DNA fragments move towards? DNA fragments are negative in charge, so they move toward the positive. All right, which one is largest of these? 2 kilobase pairs, 3,000 base pairs, or 55 base pairs? This one, because this one is 2,000. So if you're taking the test and you need to just write it out just to re-verify that you're understanding everything, just write out the freaking numbers and save yourself the headache of trying to translate it in your mind, okay? So that's why 3,000 base pairs is the biggest. All right, and how can a single nucleotide polymorphism be so dangerous? Because an amino acid can get changed, and the change in amino acids can change the whole protein structure and function. And that's it. All right, any questions? Anybody online have any questions? All right, then that is it for today. We do not have lab for today, so we're not going to be meeting today if you guys didn't get that announcement. And so that is that, and I will see you guys maybe on Tuesday. We've got snow coming in on Tuesday, right? Right. It's supposed to be a lot colder, and there's so far supposed to be more snow, but we'll see if that stays, right? We know how Oklahoma goes. So I'll keep you guys updated on that. If we need to delay a lab or something like that, I'll let you know. Okay. All right. Where is my other?